is taste bud. A taste bud shows onion-shaped structure like this, which contains about 50 to 100 taste cells. Taste buds are located on the three types of taste papillae on the surface of the tongue. The anterior part of the tongue, fungiform papillae. In the posterior part of the tongue, circumvallate papillae. And in the middle side part of the tongue, 48 papillae are located. The taste cell possess microvilli at the taste pore in the tongue surface, which can bind chemical compound, then leading to the activation of the taste receptor cell, and then the signal are transmitted to the taste nerve and conveyed to the brain. Taste bud is composed of a heterogeneous population of taste cells. 1, 2, 3, 4 types. Taste cells are classified into the 4 types according to their morphological features. Type 1 cell. This is a dark cell, considered as a glial-like supportive cell, which shows mucous circulation, cell-cell isolation, neurotransmitter degradation, and uptake by using ATPase and GLAS. These are modulation of neurotransmitter. And type 2 cell, this is a light cell. This cell have the taste receptor T1R1, T1R2, T1R3, and T2Rs. These are sweet, umami, or bitter taste receptors, and also expressed signal transduction related molecules such as the G-alpha Gustducin, tryptophan-5, and PLC beta 2. Interestingly, they don't have conventional synapse structure, even they are receptor cell. The type 3 cell, also light cell, express sour receptors, sour taste receptor autopetolin 1, and synapse-related molecules such as serotonin, SNAP25, GAD, and Angill. So these cells are considered to be a sour cell. The type 4 cell, this is a basal cell. Here, expressed develop related molecules such as the sonic hedgehog and LGR5. So these cells are considered to be a taste stem or progenitor cells. This is a picture of human tongue. That if you try to color the fungiform papillae, you can see the distribution pattern of the dot like this. You can see the many dots at the tip of the tongue here, which may strongly imply that taste organ works as a detector for nutrient or toxins before intake of food. Regarding taste pathways, the taste bud cell located in fungiform and circumvallate papillae are innervated by the chorda tympani nerve. Fungiform papilla innervated by chorda tympani nerve, and vallate papilla innervated by glossopharyngeal nerve. And the taste buds are also located in the soft palate and pharynx. So these are innervated by gracious facial nerve and vagus nerve. This test signal conveyed to the solitary nucleus, thalamus, and then finally, gustatory cortex. Here, the 5 basic test quality were recognized here. The test information further conveyed to the amygdala and hypothalamus, which regulate feeding behavior and preference and emotion evoking. In addition, at the level of brainstem, the test information induces reflection, such as the saliva secretion, gust facial response, digestive juice secretion. For example, when you eat ice cream, you recognize it's sweet here and tastes good here, and eat more here and chew more here with a big smile. And increase of saliva and digestive juice help it to be absorbed into the body. On the other hand, if you eat toxic food, you recognize it's bitter here, taste bad here, and don't eat and don't chew with unpleasant face. And reduction of saliva and digestive juice help it not to be absorbed into the body. This panel shows the gust facial responses. It's reported that taste reception system is formed by certain weeks at the fetal period. Newborn baby can respond to sweet, sour, and bitter taste stimuli. For example, the baby shows crinkling the nose and puckering the mouth to sour taste, like this, and gaping and opening the mouth to bitter solution to try to ejection the solution, like this. While as baby looks happy, there is no clear response to sweet stimuli. Babies start to drink their mother's milk from when they are born. At the moment, they are trying to determine if it's possible to drink the milk by using their taste sensitivity. From this, you can find that taste is a very important senses for surviving. To summarize the taste function so far, when we eat food, it tastes good. We will take it. But if it tastes bad, we will avoid to eat. So, well-known function of taste sensitivity is identification of needed nutrients. Furthermore, it has become clear that taste sensitivity has also systemic functions. Taste information is conveyed to the brain and then which induce facial expression, choosing, saliva secretion, digestive juice secretion, and hormone secretion, such as insulin. The hormone can modulate the taste. This help digestion and absorption. Then induce physiological function or nutritional function. So the sensitivity contribute to maintain nutritional homeostasis. So if your taste sensitivity is disrupted caused by aging, illness, or irregular lifestyle, these physiological function couldn't be induced well. So disruption of taste sensitivity may lead to diet-related diseases such as obesity, diabetes, hypertension, and malnutrition. The topic to discovery of taste receptors. The key molecule to detect the needed nutrients is taste receptors. When I started the taste research at Kyushu University in 1999, about 20 years ago, almost all taste receptors had not been identified yet. However, nowadays, as shown this slide, 5 basic taste receptors have been identified. Sweet, T1R2 plus T1R3 heterodimer. Umami receptor is T1R1 plus T1R3 heterodimer. Beta receptor is T2R, GPCR. Salty receptor is ion channel type receptor, epithelial sodium channel, ENaC. And sour receptor is autopetry 1, OTOP1. This is also a channel type receptor. So in this topic too, I'd like to trace the history of discovery of this taste receptor and its functions. The first discovered taste-specific molecule was G-protein Gustducin. Gustducin is a G protein that works with GPCR. Maybe these words G protein and GPCR are unfamiliar with you, so I will explain it. A G protein coupled receptor, GPCR, GPCR, also known as the 7 transmembrane domain receptor, is cell surface receptor that bind ligand such as neurotransmitter and hormone at the outside of the cell and induce cellular responses via coupled G protein. Ligand bind to the outside of the cell, which activate the receptor and induce the cellular response via G protein. G protein and G protein are coupled here. And ligand can bind either to extracellular region and also the bind site within the transmembrane domain. Back to the Gustducin. The expression analysis of Gustducin revealed that this Gustducin is selectively expressed in the subset of test bus cell, but not in all test cells, like this, predicting that Gustducin may mediate a certain test quality, but not all test qualities. In addition, Gustducin is a G protein. Thus, it was speculated that co-working with GPCR would be a taste receptor. To investigate the function of Gustducin, Gustducin gene deleted deficient mouse was generated. A gene is knocked out and examined how is the gastric nerve responses. As a result, the nerve responses to beta compound denatonium is reduced to almost half in the Gustducin deficient mouse. This is the wild type. This is the Gustducin knockout mouse. Such a reduction was also observed in the response to sweet substances, sucrose. Such a reduction was not observed in the other test quality, salty and sour. These results reveal that the Gustducin plays a role in mediating the beta and sweet signal transduction in test cells. In 1999, two GPCR T1R1 T receptor 1 and T1R2 T receptor 2 were discovered. These were the first candidate for the taste receptors because they are the taste-specific GPCRs. This T1R1 and T1R2 has 7 transmembrane domain, 1, 2, 3, 4, 5, 7, and long extracellular region. Here, that is likely bind taste substances. However, the function of T1R1 and T1R2 was still unknown. Next year, 2000, GPCR T2R beta taste receptor discovered, which were characterized by a short extracellular regions. Very short. Here. The expression analysis revealed that T2Rs were expressed in a subset of taste bud cells as shown in green. T2Rs. And red is Gustducin positive cells. And these figures were overlaying two images for T2R and Gustducin. And lower were T2R and T1R1. Yellow signal indicate overlapping cell of these genes, T2R and Gustducin. As you can see, most were yellow, suggesting T2Rs are co-expressed with Gustducin. On the other hand, no yellow signal were detected in between T2R and T1R1, suggesting T2R were never co-expressed with T1R. Furthermore, T2R coupled with Gustducin. So T2R would be a sweet or bitter taste receptor because Gustducin mediates bitter and sweet signal transduction, as I mentioned. To examine the function of T2Rs, newly developed functional analysis was performed. Bitter taste receptor candidate genes are introduced into cultured cells to create artificial taste cells. Then apply various taste substances and examine the cell response. Like this. So higher peak means higher cellular activation, which is followed by the receptor activation. So cellular activation indicates this T2R receptor function as a receptor for a certain ligand. This is a movie of this experiment. Can you see? You can see the clear response. Using this system, it was revealed that the cell with mouse T2R5 and T2R5 responded only to cyclohexamide beta compound, SIC. This is a response, but not to other beta testant, prop, or denatonium, no response. Similarly, the cell with human T2R4 respond only to denatonium, but not to other beta test compound. So indicating human T2R4 is a beta receptor for denatonium. Such results have been accumulated, and it's now clear that there are about 25 types of THRs function as a beta taste receptor in humans. Again, THR family is characterized by the 7 transmembrane domain with a short extracellular region and bind only test substances with a specific structure. For example, THR4 is for denatamium, THR38 is for PTC. These results also suggest that for surviving, many receptors were required to detect various beta toxic compounds in the evolutionary process. In 1999, two GPCR, T1R1 and T1R2 were discovered, but their function was still unknown. 2001, of these, T1R2 was revealed to function as sweet taste receptor combined with a new member T1R3 combined. How do you think about the method to reveal the function of this T1R2 and T1R3? I suppose you can answer. As you supposed, the taste receptor candidate gene T1R1 are introduced into the culture cell to create artificial sweet taste cell, then applies various sweet taste substances and examine the cell response. As a result, when each T1R2 or T1R3 were introduced into the cell individually, the cell did not respond to any taste stimuli. But when both T1R2 and T1R3 were introduced together, they showed clear responses to sweet compound. Thus, these results demonstrated that T1R2 and T1R3 makes a heterodimer and function as a sweet taste receptor. Regarding sweet taste sensitivity, there is one big question, that is, the chemical structure of sweet substances very vary. For example, natural sugars, glucose sugars, like this, artificial sweeteners, saccharin, aspartame, cyclamate, are like this, and sweet proteins, neocolin and monoline are so very big molecules. And amino acid, sweet amino acid are very tiny molecules. Despite the lack of common structure, every compound induces sweet taste. What is the molecular mechanism? As I mentioned, a characteristic of these T1Rs has a long extracellular region. And T1R2 and T1R3 is a heterodimer. So this has two long extra region, 1, 2, and 2 transmembrane domain. So it was speculated that sweet taste receptor would have multiple ligand binding site like this. To examine this possibility, T1R2, T1R3 mouse human chimeral receptor were generated and analyzed. For example, human and mouse sweet taste receptor are shown in the red and blue, which were separated into the three regions: extracellular region, system-rich region, and transmembrane region. And only the extracellular region of T1R3 is replaced between human and mouse, like this. T1R3 MHH means extracellular domain is the mouse, and system-rich domain and transmembrane domain are human. This is the human mouse chimera T1R2 and T1R3. By using this chimera, sweet taste receptor and usual artificial taste cell, ligand binding site for various sweet taste substances were investigated. The chimera. Then now many results have been accumulated and revealed that T1R2 and T1R3 has multiple binding sites. For example, binding site for artificial sweeteners, aspartame, natural sugar, sucrose, glucose, and sweet amino acid tryptophan bind to the extracellular region of T1R2 here. And sweet protein, neocolin, bind to the extracellular region of T1R3 here. And the sweet protein monelin bind to the cysteine-rich domain of T1R3. And artificial sweetener cyclamate bind to the transmembrane domain of T1R3. Now we know the answer for the question about the existence of various sweeteners. That is, there are multiple ligand binding sites on T1R2 and T1R3, thus which can bind various sweeteners with different structures. Furthermore, in the process of evolution, sweet taste receptor may have evolved by increasing the number of ligand binding sites so that various nutrients can be detected in order to enlarge our body and brain. It was revealed that T1R2 and T1R3 function as a sweet taste receptor, but the function of T1R1 has been remained unknown yet. In 2002, it was revealed that this T1R1 made a dimer with a T1R3. This is also C2 receptor component and function as umami taste receptor by using usual artificial taste cells. Umami is a Japanese word, and umami can be translated as a pleasant, savory, or meaty in English. Glutamate is a typical umami substance which was discovered as a delicious ingredient in kelp by Japanese researcher Dr. Kikunai Ikeda about 100 years ago. It's also known that glutamate is abundant in mother's milk. So breast milk is so umami for babies. Inosine monophosphate, IMP, and guanosine monophosphate, GMP, are known as other umami taste substances. And they are abundant in dried bonito or shiitake mushroom. The characteristic of this umami taste is synergistic effect. Synergistic effect means mixing glutamate with a small amount of nucleic acid, IMP or GMP, enhance umami intensity. This is a raw data of the taste nerve responses to MSG alone, glutamate alone, and IMP alone. And this is the MSG plus IMP mixture. So as you can see here, larger response were observed in the mixture than in the sum of MSG alone and IMP alone. Such synergistic effect is observed only in umami taste quality, but not in other taste qualities. Regarding the molecular mechanism underlying this synergistic effect, it's supposed that the binding region for glutamate and IMP were different in the ligand binding site of T1R1 extracellular region, which looks like the opened mouth of the Pac-Man game character. Glutamate is outside here and IMP bind to inside. And this opened mouth is closed more tightly by binding of both MSG and IMP. This is a Japanese soup stock, dashi. Dashi is made by heating water containing mixture of kelp here, dried bonito here. So as you found, the synergistic effect is applied in this Japanese dashi. Dashi is used to add umami to not only soup but also to various Japanese dishes such as tempura and shima's dishes in Japan, like this. These traditional Japanese cuisine, washoku, utilizing dashi. This was recorded on the UNESCO's list of the World Intangible Cultural Heritage in 2013. Salty and sour tastes are induced by sodium or proton ions respectively, so they are supposed to be a channel type receptor. Regarding the salty taste, it has been well known that diuretic amiloride, this is the drug, suppressed salty taste response. A diuretic is a substance that promotes production of urine. So the salty receptor has been expected to be an epithelial sodium channel ENaC because ENaC is suppressed by this diuretic amiloride. Based on this hypothesis, taste-specific ENaC knockout mouse was generated and examined their taste responses. As a result, amiloride-sensitive salty taste component was specifically disappeared. This is a controlled mouse, and this is a ENaC knockout mouse. The orange part has completely disappeared. Thus, this result revealed that ENaC is a salty taste receptor. Lastly, regarding sour taste receptor, its identification was very difficult because there was no molecule modifying sour taste, unlike the Gustducin for beta or sweet receptor or amiloride for ENaC. However, in 2018, finally, otopetolin-1 was discovered as a sour receptor by a comprehensive analysis of gene expression in type 3 sour responsive cell. This is a summary of topic 2. The receptor for sweet umami beta are GPCRs. When these GPCR are activated, G protein, can you see? Blue, blue. And downstream molecules, PLC beta 2, IP3R3, and tryptophan 5 convey the information one after another like a bucket relay. and finally induce the action potential. And leading to the release of neurotransmitter, which activate taste nerve. Interesting point is that these transduction molecules are common, Gustducin, PLC beta 2, IP3R3, among these taste signals. The taste receptor for sour and salty are channel type receptor. A proton and sodium ion pass through their receptor to between 1 or NaCl, which also induce action potential and leading to the release of neurotransmitter and activate the taste nerve. Now we know all taste receptors for 5 basic tastes and their transductions. The topic 3, extraoral taste system. Surprisingly, taste receptor are now known to be expressed in multiple organs and serves as a sensor for different functions among the organs. Yeah. For example, hypothalamus regulating food behavior express T1 node 1, 2, 3, which may relate to energy sensor. The pancreas is important to secrete the insulin. Pancreas expressed T1 node 1, 2, 3. This regulates insulin secretion. The salivary gland and kidney expressed the NaCl salty receptor, which regulates sodium reabsorption. The trachea expressed T2R beta taste receptor, which are related to the antibacterial immune system. Lymphocytes also express T1 nr3. This is also related to immunity. Gut is very similar to the oral taste bud, express almost all taste receptors and regulate the hormone secretion and nutrient reabsorption. Testes also express the T1R or T2R, sweet umami or beta taste receptor, which affect the sterility. Collectively, the taste system expressed in multiple organs may be involved in multiple functions, and thereby this may be involved in the multifunctional failure, such as obese diabetes, hypertension, hyperlipidemia, infection, infertility. So by examining the further about the molecule and the function and gene analysis, single nucleotide polymorphism and diseases, the function in the various organs, and using new special technique, this is a test bud organoid in vitro organ 3D-dimensional culture system, new technique. By using this examination, we and you can contribute to human health by understanding of the function of taste signaling and define the broad significance of the taste system in humans. The topic 4 is the taste renin-angiotensin system may contribute to the maintenance of sodium homeostasis. This is our research work. The subtitle is Possible Linkage Between Taste Renin-Angiotensin System and Taste Loss in COVID-19. This topic about salty and salty modulation is based on these our papers. The first part, angiotensin to modulate salty and sweet taste sensitivities. Firstly, I'd like to talk about the background of salt taste study, our salt taste studies. Before starting the research, the big question is whether salt taste sensitivity is modulated. A long time ago, it has been reported that adrenal ectomystrate sodium deficient in the body, mouse with very low blood sodium level, mouse prefer to drink very high concentration of sodium chloride solution. They usually reject. So please imagine that you drink the sea water? Maybe you cannot drink, but they like it, which is specific sodium, natrium, magnesium, and aluminum. Moreover, nerve responses to sodium are suppressed in such sodium deficient rat. From this result, we speculated that the salt test sensitivity is modulated by unknown humoral factor depending on blood sodium level. It's well known that sodium homeostasis is regulated by renin-angiotensin system. Angiotensin 2 is produced by renin and angiotensin converting enzyme 1 ACE1 from angiotensin organ. And angiotensin 2 induce aldosterone circulation via its receptor 81. Aldosterone stimulate sodium reabsorption via epithelial sodium channel ENaC, which result in the increase of body sodium level and blood pressure. Interestingly, angiotensin 2 also induce sodium appetite and ingestion acutely. However, the molecular mechanism involving salt signaling is unknown. So we hypothesize that if angiotensin 2 receptor 81 is expressed in peripheral T cell, circulating angiotensin 2 may also act as a modulator for amilor sensitive salt responses, which cause increase of sodium intake. First, we examine the expression of messenger RNA for angiotensin 2 receptor 81 in mouse test bud cell by RT-PCR and in situ hybridization. FP, VP, ET means fungiform papilla, Barrett papilla, test papilla, and epithelial tissue without test bud. The results show that AT1 angiotensin II receptor are expressed in subset of T cell here in both fungiform papilla and Barré papilla, fungiform papilla and Barré papilla, not in epithelial tissue without T spot. These expression patterns are very similar to those of T cell marker T1R3, as you know already. Sweet or umami T cell receptor component like this. These results suggest that angiotensin II may be able to act on peripheral T cell via AT1. Next, we asked whether angiotensin II modulate T nerve responses in mice. It's known that there are two salt T receptor pathways. One is amyloid sensitive AS salt receptor pathway, which is mediated by ENaC. You already know AS pathway. The other is amyloid insensitive AI salt sensitive pathway. And both AS and AI were observed in anterior part of the tongue innervated by chorda tympani nerve. So we focused on the CT nerve responses containing both AS and AI current. The results show that CT nerve response, chorda tympani nerve responses to sodium chloride were significantly reduced after administration of angiotensin II. The peak is lowered, reduced, suppressed. This is a control. This is no change. Only sodium chloride is reduced. This response contains AS and AI. The nerve responses to sodium chloride and amiloride, which elicit only AI current. The amiloride block the AS, so this response from AI. This AI response was not changed. Furthermore, surprisingly, response to sweetener glucose sucrose saccharin was significantly increased after angiotensin II administration. Response to other test substances such as savory, Umami, MSG, and quinine beta are not changed. These results suggest that angiotensin II selectively reduce AS salt responses and also enhance sweet taste response. Next, we looked at the co-expression pattern of AT1 and T cell receptors. The analysis demonstrated that AT1 receptor are co-expressed with ENaC sortase receptor. And T1R3 sweet umami receptor, as you can see here in the yellow, are observed. But AT1 is not co-expressed with PKD2L1. This is a savory receptor candid gene. You cannot see the yellow signal. It has been reported that 5 basic T receptors co-expressed in 5 different T cell populations. Thus, angiotensin II induce salt and sweet taste modulation may be occurred independently through distinct taste cell expressing ENaC or T1R3. AS cell express ENaC or sweet cell expressing T1R3. AT1 here co-expressed with T1R3 co-expressed with ENaC here. I would like to summarize first part. Our results suggest that when body sodium level is lowered before meal, blood angiotensin 2 level is increased, which suppresses the sodium test sensitivity, causing increase of sodium preference and intake. And then body sodium level is recovered, angiotensin 2 reduced. Then salt test sensitivity is recovered, causing reduction of sodium preference intakes. The regulation of angiotensin 2 on peripheral salt test sensitivity would contribute to salt intake and play an important role in sodium homeostasis. Furthermore, the angiotensin 2 also enhance sweet test responses, which contribute to increase of calorie intake. The linkage between salty and sweet taste preference via angiotensin 2 signaling may optimize sodium and calorie intake. The second part, renin-angiotensin system, LAS component in the taste organ may contribute to regulate taste sensitivity acutely in mice. It's now recognized that various tissues have an organ-specific RAS. For example, renin-angiotensin aldosterone, RAS again, a renin-angiotensin system. RAS component, renin-angiotensin again, ACE1 are expressed in the kidney. And angiotensin 2 is produced within the kidney and functions as various function. Similar results were observed in other various organs. These findings raise the possibility that organ-specific RAS might also exist in taste tissues, and angiotensin 2 might be produced locally here in response to oral environment. First, we examine the expression of messenger RNA of renin-angio system component in test bud cell by RT-PCR and in situ hybridization. Renin-angiotensinogen and angiotensin converting enzyme 1, angiotensin converting enzyme 2. Of these, renin-angiotensinogen and ACE1 are expressed in subset test cell in test bud, like this. Renin, here, angiotensinogen, like this. Angiotensin converting 1, this is a control, T1R3, and this is ENaC. Angiotensin converting enzyme to ACE2 were detected in all tissues, Fanny-Home, Barrett, epithelial tissue with a test bud. These results strongly suggest that all renin-angiotensin system components were expressed in mouse taste bud cell in both FB, Fanny-Home, and Barrett papillae. Next, we perform the co-expression analysis for last components. Renin positive cell co-expressed with T1R3, like this, yellow observed, in both fungiform papilla and Barrett papillae. But renin did not co-express with GAD. This is the savory receptor marker. Savory receptor marker did not co-express with this GAD GFP. Renin positives also expressed NaCl sortase receptor and 81. Renin is co-expressed with 81. 81 is the angiotensin 2 receptor. Yellow, yellow, yellow. I also examined about the angiotensin organ and ACE1. The expression is shown here. This is a summary of co-expression analysis. As you can see here, all last component and 81 are co-expressed with T1R3 or NaCl in test bus cell. These results suggest that the test organ contains a local RAS, which may be capable of producing angiotensin 2 and receiving it within the test bud. I would like to summarize second part. Our results suggest that test organ has a taste-specific RAS, which can produce angiotensin, produce, and receive via 81 within the taste bud in autocrine or paracrine manner and modulate both salty and sweet taste sensitivity. And then, after meal, angiotensin 2 is rapidly degraded by ACE2, which may contribute to short-term regulation of salt test sensitivity. This is the summary of part 1, first part and second part. The specific modulation of amiloride-sensitive salt and sweet test sensitivity may be mediated by both locally produced angiotensin 2 temporary feed-forward regulation, locally produced RAS, and circulating angiotensin 2 continuous negative feedback regulation. This is the circulating RAS. Mediated by both RAS. Such an integrated regulation of peripheral test sensitivity by angiotensin 2 may play an important role in sodium-calorie homeostasis. We speculated that this local RAS could help to avert decreases in the body sodium level or prevent excessive sodium calorie consumption independently of changes in body sodium chloride balance. The third part is a possible linkage between test renin-angiotensin system and COVID-19. Now we are in COVID-19 pandemic. As you know, it has been reported that new coronavirus SARS-CoV-2 infection causes taste and smell loss. In the early stage of the viral infection, SARS-CoV-2 utilizes its spike protein to bind to the host cell membrane via ACE2. ACE2 is the attachment receptor for S protein, the spike protein. Then virus can fusion with the host cell following the spike protein priming by transmembrane protease serine 2 template 2. This is a protein priming enzyme. So ACE2 and template 2 were required for SARS-CoV-2 infection. This is reported by Cell to 2020, April. Before publishing this Cell paper, we had reported the expression of ACE2 in the test bed as mentioned in the second part here. So now this nutrient paper has attracted a lot of tensions. So I further examined the expression of ACE2 and template 2 by using our gene chip data. Tissue sample were test bed from fungiform papilla and Barrett papilla and ganglia from geniculate ganglia and nodose petrosal ganglia and trigeminal ganglia, which contain the chorda tympani nerve neuron, taste neuron. This also include the taste neuron, glossopharyngeal nerve neurons. Trigeminal ganglia don't have the taste neuron without taste neurons. This is a negative control. Looking at the expression of ACE2 and template 2 in the test bed of fungiform papilla and second Barrett papilla, you can see that ACE2 messenger RNA were highly expressed in both fungiform papilla and second Barrett papilla. PPP means positive, positive, positive. Three data were from different gene chip probes. Similarly, template 2 messenger RNA also positively expressed PPP. Next, looking at the data in the three ganglia, ACE2 messenger RNA were negatively expressed in the all ganglia. AAA means absence. Similarly, expression of template 2 was also very faint. AAPA. This is a taste ganglion. This is ganglion. These results suggest that taste disorders in COVID-19 patients may be in part caused by direct infection of COVID-19 to taste bud cell, but not to taste neurons. Via is to template 2 expressed in taste bud cell. I also examined the expression of ACE2 and template 2 by using RNA-seq open data by other research groups. This is a Nature paper. This is a scientific report papers. And they examined the expression of these genes in beta cell specific or sweet cell specific. Then you found that expression level of ACE2 and TMPRSS2 were very low in both beta and sweet cell, but not negative. TMPRSS2 also expressed in both beta and sweet cell. The other single-cell RNA sequence study showed ACE2 messenger RNA expressed in about 30% of type 3 cell. These are each are a single sour taste cells, and expression were examined. This is one cell, one cell, one cell. The is still expressed in some of sour taste cells. So 30% are expressed. The S2 were also observed in T1 now 3 positive cell. This one is a sweet or umami taste single cell. But temperatures 2 were not observed in both type 3 or type 2 cell. So these study also showed ACE2 and TMPRSS2 expression in taste bud cell. The third study indicate that ACE2 and TMPRSS2 were expressed not only in some taste bud cell, most are in non-taste epithelial cells of the filiform papillae. Red is the ACE2 positive cell. They showed that ACE2 is expressed in the very small amount of the cell. Amount of expression was observed in test, but also in the many most the basal cell of filiform papillae. This is a non-taste epithelial tissues. So SARS-CoV-2 can directly bind and infect to the disease cell via ACE2. This is the last slide. It's known that dehydration, hypertension, hypernatremia, diabetes, kidney disease, Gustducin syndrome, exhibit high blood sodium level. Thus, it's also suggested that circulating renin-angiotensin system and taste renin-angiotensin system may be upregulated in this patient and in the patient with these diseases. So now we speculate that the people with these diseases may have more susceptibility to SARS-CoV-2 infection via upregulated ACE2 in the taste bud and tongue epithelial tissue cells, which may cause severe taste dysfunction, which may be due to the taste cell apoptosis or inflammation. Thus, in order to prevent taste loss, we speculated it may be important to control the loss activity in the taste bud by taking proper medication. This is our member. If you want to know us more, please check the keyword taste or Shigemura by internet. Thank you very much for your kind attention. Thank you. That's all. It's okay? I'm sorry. Uh, I'm so I'm sorry. I can't hear not anything. The bad internet connection. Maybe you now. Doctor answer speaking. I'm sorry. Can I hear? If you okay. I'm sorry. Bad connection. I'm sorry.
Bagaimana dengan sekarang? Apakah bisa mendengarkan suara saya? Bisa? How about now, Sensei?
No? Okay, now. Okay.
Okay.
Now I can hear.
Thank you. Oh yeah, thank you very
thank
much.
you. Thank you very much.
Thank you for the interesting presentation, Sensei. I believe your presentation and your topic is strongly related with the research that have been conducted by Dr. Jenny Sunariani, Professor Jenny Sunariani. So for this first opportunity, I would like to invite Professor Jenny to give some comments or maybe some questions regarding the Professor Sigamura's presentation. Professor Jenny, please.
Thank you very much for the moment, Dr. Oki. The presentation of Professor Sensei is very interesting for me because I'm my research tree is also the taste, and I was make research, many research. The one of my research is mastication, and then the research the research is up because if a baby mastication and hard food, there's no research, no significant research. But if the mastication and middle food, the result is sweetest is more more significant than the other. And then the research only gave that mastication and food, middle food, is give the baby more high cognitive. This is one of my research. I want to ask to you. I hear that you your research about the research of REN, that angiotensin gave the
Kidney
result,
maybe.
Yeah.
Kidney.
Yeah.
Kidney, not REN.
Oh yeah, the kidney. The result is angiotensin gave a significant in this your research. My research in Indonesia at the kidney gave result that if the gagal ginjal keempat itu apa deh?
Renal kidney failure.
Yeah.
Kidney failure.
Failure that the sure taste is more this is more more or less the taste of sure. I'm sorry. My English is so more. Yeah, gitu aja deh. Okay. This is my research, two of my research about the research for about the whole body.
Gimana, Prof Jenny?
Ya, saya sampaikan kalau saya itu aja yang saya ceritakan pada dua penelitian saya pada terhadap tubuh, yang lain tidak saya ceritakan. Nanti akan membuang waktu. Ada dua penelitian.
It's not a question ini, it's just comment.
Yes.
It's not a question.
Nah, saya ingin menanyakan kalau di Ando itu kan beliau kan menyatakan ada hasil manisnya ya, ada hasil manis pada penelitian angiotensinnya tadi, ginjal. Tapi kalau yang saya ketemukan adalah rasa pengecap asam pada ginjal itu menurun. Demikian juga pada orang yang mencium bau bom itu juga terjadi penurunan rasa asam. Itu aja. Masih banyak penelitian saya kan, tapi
Yep. I will try to translate Professor Shigemura. According to Professor Jenny's studies, she found that in the case of kidney failure, there is some or many cases of loss of taste of sour. Not sweet,
sour.
Not sweet taste, but sour taste. Maybe it's a little bit different with your findings, but that is what she found in her studies.
That's very interesting.
Okay. Do you have any comment about it?
So I don't know the reason, but I don't know exactly the research performed by Professor Jenny. And I asked that.
Okay.
I'm sorry.
Yeah, okay. Thank you very much. Now, I give the next opportunity to the student. I believe some of the students have a question. Please, for those who has the question, can deliver the question to Professor Shigemura. Please, the student who has question, please ask Professor Shigemura in personal right now. Josephine?
Yes, doctor. Okay. Thank you for the opportunity, Dr. Aksa. And I have a question for Professor Noria. So, the presentation pages, I've seen some of research-based use are already past more than 5 years. Is it still help.
I'm sorry. I couldn't
Yeah.
hear.
I think, Josephine, we have some difficulties to hear your voice.
Radio, okay, doctor.
Or you can post your question through the chat that Professor Shikimura will read the chat.
A chat? Okay, I will check.
Yeah.
So from the morning,
Yeah.
here, one question. So first question is, I want to ask the question, if test receptor dependent on channel protein, G protein coupled receptor along with its ligand, does the perception of smell play a role in perception and smell? Little bit very difficult question. So, COVID-19 affect the you think the smell is the main reason of the this loss of the smell or taste perception. The smell loss related to taste perception? No. The question is,
Okay.
but so very difficult to so here and talk. So I cannot understand the question. Maybe I'm sorry. So can you hear? Can you hear me? Now can you hear me? Okay. Okay. I try to answer. And the smell loss may be caused by the direct infection of SARS-CoV-2. The host cell is around the cell. That this is not the olfactory receptor cell, not olfactory cells, the around cell. So directly infected to the nasal epithelium. So this is also revealed that the reason why the taste smell losses occurred. But the taste loss, the reason of taste loss is not revealed yet. My presentation is one possibility. The ACE2 is expressed in taste buds, so SARS-CoV-2 can infect the taste cell directly. And we feel that taste is both from the taste and smell. If you like this, stop the smelling, you can't taste well. So both smell and taste is important to feeding behavior. So smell loss may related to the taste loss. I'm so good.
Yeah.
Okay.
Yeah.
The second question is, excuse me, your sister, I have a question. On the previous presentation page, I have seen some of research-based use already past more than 5 years is still valid if used in present time. More than, you know, Because as we know, scientists will always be developed as process science technology depend on error.
Mungkin yang dimaksud hubungan antara semangates ya.
Yeah, I couldn't understand the question but this is a very correct comment, I think.
Yeah, they are the first grade students. Maybe they have their perception is the reference is better not more than 5 years. Yeah, and they found that in your presentation you put some some reference that 4 than 5 years. Then they ask about it.
I think that the result is not changed. This is correct. I believe it. And the technique is difficult, different, but the result is not changed, I think. Because we speculated a very long time ago that ENaC may be a soltase receptor. So, and after that, and about 10 years ago, 10 years later, and 2010, ENaC is revealed via a soltase receptor. We hypothesized that the ENaC is a receptor, and then revealed this is a real soltase receptor.
Ya, jadi gitu ya, Sevin ya. Jadi kira-kira 5 tahun salah. Okay. Is it clear? Is it okay, Sensei?
Yeah.
It is finished?
Okay.
Yeah. We cannot hear anything from you, Josephine. I'm sorry. Any other question?
May I ask some question for the professor?
Yeah.
Is my voice clear?
Yeah, it's clear enough. Hopefully, it still remain like that.
My name is Salma Najmi, and I want to ask some questions. Is there any possibilities that age and races, like Indonesians or Chinese, have different sensitivity in taste? Maybe Indonesians have more sensitivity sensitivity in salts more than the Chinese races or something like that?
Okay, Shigemura Sensei.
I'm sorry, I couldn't hear.
Yeah. She asked about, is there any sensitivity in particular tastes between one race to another race? For example, Chinese person and Indonesian people, or in the Western people.
So is there the difference in the taste sensitivity among the Asian and the European? The question is,
Yeah.
yeah. Maybe yes. For example, we have performed the experiment that showed that umami taste sensitivity. So it reported that the Western people and especially the obese and women like and more prefer to the umami substances. And also, and we examine the gene genome, the single nucleotide polymorphism for the umami receptors. And we found that the some single nucleotide polymorphism affect the umami taste sensitivity, and the percentage of single gene mutant is different among the populations. The Asian have the more SNPs, and the Western people are not so many people show the low frequency of SNPs. So maybe the gene affect the taste sensitivity among the populations.
Is it clear, Salma?
Thank you very much, Professor, for answer.
So I think very important thing is examination the more detailed of the genome data among the human populations.
Genome data. Any other question from the student and the staff? I believe this topic is strongly related with the topics that have been taught in the semester by the subject of oral biology. So I believe you have been you receive the kind of material of the taste in the semester. You must have something about the taste to ask to the Professor Shigemura. If there is no question, maybe I have a question, Sitsi, about the addiction of a particular taste. If we eat a lot of sweet taste, we want to eat more sweet taste again and again and again. We want to repeat this, to eat sweet taste day by day, tomorrow and the day after tomorrow. This is called taste addiction. How is the mechanism of the taste addiction in the brain?
Very difficult question and hard to answer. And I think this is caused by the brain system, not taste sensitivity. I think that the central nervous system is modulated by eating, the repeated eating. Maybe the neurotransmitter or hormones such as endocannabinoid or the like morphine is more circulated by such repeated feeding. So we feel more good, and so this is related to addiction. I think the brain system, not taste sensitivity, I think.
Okay, yeah, great. So it is correct that if someone stated that the center of the food addiction is the same with the center with the drug addiction. For example, narcotics. If someone has the drug addiction, or a drug addiction to the narcotics, the center of the addiction is located in the temporal lobe. Yeah, some scientists say that the temporal lobe is also responsible place to the food addiction, in particularly sweet addiction. How about your comment about it?
So I'm sorry, I cannot I can't understand it. Your question. I'm sorry. Again?
Yeah. Again, okay. In the addiction of narcotics or drug addiction, the location of drug addiction is in the frontal cortex, in the frontal cortex. Some scientists say that the center of the food addiction is the same place with the drug addiction in the frontal cortex, especially this addiction for the sweet taste. Have you heard about it, or do you have any comment about that fact?
This is also hard question. This is a central nervous system. So I mainly examine the peripheral taste this area.
Okay.
So I don't know the reason why the exact price is. So maybe you know more very much. I think. Right.
Thank you very much. Any other question from the student and from the staff? I believe everything that has been delivered by Professor Sikimura is very valuable. It is very important for us to enrich our knowledge in the field of oral biology and in the field of oral physiology, especially for the staff who has extend their knowledge like Prof. Yeni in the area of the taste. I think it's very important, and we wish this activity will be, proceed with some kind of collaborations, and say our staff will have collaboration in research about the taste, yeah, with your institution. Webinar is almost finished. In the attention. Thank you to especially to Prof. Jenny for the comments. And webinar maybe next year we will invite you again. Thank you very much for everybody. Bye-bye. Have a new year.
Thank you. Bye.
Goodbye.
Thank you, Professor. Thank you, Professor.
Thank you, Doctor. Thank you, Prof.
Thank you very much.
Thank you, Doctor. Arigato, Sensei.
Sudah selesai ya. Ini sedang proses selesai. Monggo di lift saja Bapak Ibu.
Yeah.
Saya izin live ya.